Deep within the microcosm of the human body, the most complex piece of microscopic machinery resides, a cell in its natural habitat. This cell is about to undergo a process that will not only drastically alter it, but will completely redistribute its components. This vital process is called mitosis. Over the course of its lifetime, a cell exists in many different phases. 7 out of the 8 phases and subphases last for mere moments. The cell is most often found in interphase G1, where it goes about its daily business self-regulating.
The first inkling of change happens where mitosis begins, in interphase 2. Interphase 2 sees the appearance of a new centrosome, not unlike a tiny sea urchin. The centrosome has small tubes called microtubules that emerge from it. From here, the cell quickly moves on to prophase, where we see the biggest change. The spaghetti-like mass of genetic material that was at the center of the cell has now condensed into chromosomes, tightly packed X-shaped bundles of DNA. The halves of each chromosome are known as sister chromatids, which are joined at their center by a centromere. Meanwhile, the centrosomes have not been idle, lengthening their tubules in order to form what is known as the mitotic spindle.
Then quickly on to prometaphase, where the nuclear membrane has dissolved, allowing the rapidly growing spindle to attach itself to the chromosomes via locations called kinetochores. The other tubules have joined together across the cell, anchoring the centrosomes into place. In metaphase, the purpose of the spindle fibers becomes apparent, as motor proteins begin shortening the tubules attached to chromosomes, moving them all to the center of the cell. The chromosomes are lined up along an imaginary line in the center of the cell, known as the metaphase plate. This comes in handy during anaphase, which splits all of the chromosomes down the middle and begins pulling the halves in opposite directions. The force of this division is so great that it causes the entire cell to begin stretching in the direction of the motion.
Each chromatid contains the amount of genetic material that a healthy cell needs, so in telophase, the prepared chromatids are simply given new nuclei. Now back within the protection of the nucleus, chromosomes begin to dissolve back into their spaghetti-like form. There is also no need for the mitotic spindle any longer, so it too begins to decay. This process concludes with the final stage, cytokinesis. A cleavage forms between the nuclei, splitting the new cell completely. There is now one copy of every vital organelle in each new cell. In effect, the cell has been cloned. This concludes one of the most intense series of changes to take place in this microscopic level.